Hello, everyone. So today we are having a next lecture, and while it's loading, I thought, how do you transition from the previous one when we were talking about the tissue engineering and technologies like this? And yeah, and it turned out that it's not so hard because recently a paper was published about some organoids that were made from neural tissues. And we discussed organoids last time, but it turns out that some organoids are not like others, in the sense that if you make organoids from human brain, if you induce stem cells to make them neurons, they can create very sophisticated structures. And it turns out that these human brain organoids, they can produce coordinated waves of activity when you stimulate them. And what scared researchers in this case is that these waves are very close to what we can see in premature babies when you stimulate their brain. So in this case, people thought, have we really gone too far? Have we created something that we cannot really control in lab settings? And while these organoids are very useful models for neuropathic diseases like depression or autism, it turns out that it might be not exactly ethical to grow them in lab settings. So, and I would like to start this lecture with this problem. And the very interesting topic that they discussed in this paper was about the consciousness. So they thought that these organoids are in fact conscious because they can produce these coordinated waves of activity. So for a bit, we are going to be talking about how do you find traces of consciousness in these neural systems. So, and to do this, to analyze these types of systems, we need to talk a bit about features of neural systems that are probably known to you. So you may know that neural system is characterized by excitability and conductivity. And it turns out that science is interested in both of these characteristics. So we can study both functional information in the sense that we want to know what gets activated when. And we also want to know the connectivity information about the conductivity part. So we would like to know what interacts with what in neural systems. And yeah, I'm going to be doing some strange stuff between slides because for some reasons they're lagging behind for the presentation. So right, I don't know why. So we are now on the first slide, and some reasons yes. The, all right, so we managed to get luck under control for some reason. So let's first talk about the functional part of what we want to know about neural system. And for this, let's talk a bit in very minuscule details, in very common terms about the functional magnetic resonance imaging. It's a very common technique. And the underlying principle there is that it tracks changes of oxygenated blood flow. And it could be taken as a proxy for neural activity, because if you have, or if some part of your brain is excited at this moment, if it processes something, then you may say that this part of the brain will be better oxygenated. Of course, you should take into account some temporal lag, in the sense that first it has to be excited, and then you would expect the increase in the blood flow. But all in all, you can study this type of parameter and make assumptions of about the functional characteristics of the brain. And so it has about 1 second of temporal resolution and about 1 millimeter to the power of 3 is a spatial resolution. So it is a very it's not exactly precise in terms of both types of resolutions, but as for temporal resolution, it's very good and it can be used in a lot of settings. So, and it turns out that functional magnetic resonance imaging is used for a lot of things. And so there are a lot of medical uses for this type of technique because while you might not be always interested in the excitability of the brain, it can still be used to detect brain injuries, tumors, ischemic regions. So this stuff, especially ischemic regions, are directly correlated with the blood flow. So if you want to make some decisions about the surgical intervention, you may rely on this data from fMRI. So you can use this as a planning for operation and diagnostic tool. And the reason, one more reason for the usage of fMRI for planning for operations, and the reason for this is that our brains are not exactly alike. So human brain is anatomically variable, and there are some hotspots that are especially variable. So you can see that colors that are more resemblant to we are having some internet connection problems. All right, connection restart. So, so there is a reason why you may want to perform fMRI prior to surgical intervention, and the reason for that would be that human brains are anatomically variable, and there are some parts of the brain that are more variable than others. So you can see that the ones that are more more akin to red, they are more variable. And in this particular study, people looked into this brain-to-brain variability between different species in humans and in monkeys, not exactly this one, but it turned out that variability is also, while it is region-specific, it can be connected with brain functionality. For instance, in humans, some variability arises in regions that are connected with the articulation of speech, and in some parts of the brain that were extended during the human evolution. So we are reliant in a lot of senses on our brain for our evolutionary success. So it turned out that this brain-to-brain variability is connected with these regions in the brain. So, but if you are planning the surgical intervention, that may spell a problem for this, because you don't really know what to upgrade if you do not know the correspondence between the functionality and the anatomical structures. And for this, you might perform fMRI. And for this exact reason, also, people created brain atlases that serve as reference materials for comparison between brains. So our brains are not exactly alike, but we can say that some area within the brain, some gyrus or some sulci are very closely resembled. They may not be in the same place within our head, but if you map their functionality, you may find the exact anatomical region where it resides. And you can also, in these brain atlases that contain a lot of these sections of the brain, you can also incorporate gene expression data for more biological resolution. So you can type different cell types, and you can be more precise in your interventions or studies. So, and while we're talking about the fMRI, it is worth mentioning that this technique was misinterpreted a lot. So there are some problems with fMRI, and one very famous example of this problem with fMRI was the study on dead salmon. So in this case, the dead salmon was shown images of people in different social situations with a predefined emotional valence. And it turned out that if you did study just right, you could find some neural correlates of different emotions within the brain of the dead salmon. And but it turned out that the whole study was really about the importance of multiple comparison corrections, and people were aware of this. But there are some more ways you can mess up your fMRI studies. So yeah, multiple corrections are really important. Multiple comparison corrections are important, so please do them. But there are also some ways you can make mistakes in these types of studies. For instance, if you misinterpreted your data, the way you can do this is to generalize it beyond measure. So you can find some kind of a neural correlate for some activity, but you can generalize it in a very bad way. For instance, you can try to interpret in a politically loaded way. You can say that if a person is looking at the picture of some candidate and you find some neural correlates connected with looking at this picture, you may think that this may be demonstrative of the person's political views and so on and so forth. So it is more of a problem with the interpreting data than the method itself. And also another problem is that for just about anything, you can probably find neural correlates, because brain is active most of the time. So if you are not dead, you will find some activity in different parts of the brain, and you can probably find some kind of correlations, but you would not, in most cases, you would not be able to say what it means. So these are some problems with fMRI. And as for consciousness that we were talking about, consciousness in organoids or consciousness in general, it turns out that some studies found places in human brain that are correlated with consciousness. And mostly these are parts of the brain that reside in the parietal lobe, on the border of parietal lobe, occipital lobe, and the temporal lobe. And most of the time you can see activity there during the states that are considered conscious. And as an interesting part is that if you manipulate somehow with the frontal lobe, so if you perform some kind of a neural surgery on in these parts, you do not really interfere with consciousness. So a lot of people that were subjected to lobotomy, they maintained their consciousness and they did not really suffer a lot from this. So consciousness was not affected in these cases. So yeah, let's go to another technique that can be used for analyzing the functional state of the neural system. And this technique, it uses a different approach to detecting the activity within the brain. The main idea behind the electroencephalography is that you place electrodes on the head and detect whatever electrical activity you may find. The good thing about this is that it is really fast, so the temporal resolution is really high, but it gives a very approximate estimate of neural activity. So you are recording the the function of multiple neurons at the same time, and also it is messed up by the fact that you record it through the skull, through all of the tissues that lie between the electrode and the neural system. And so, yeah, you can only have very approximate measures of what happens inside the brain. But still, it is used a lot. And there are some even entertaining approaches you can use electroencephalography. For instance, there are some toys that utilize very simplistic electroencephalographs. And so like this one, when you have to concentrate to make the ball float. And as for the consciousness, it turns out that there is a very interesting technique that can be used to assess the consciousness of an individual. So we do not really understand right now what consciousness is, but we can have some kind of a working estimate for this. And the technique in question is the technique that's called ZAP and ZIP. And ZAP here stands for the fact that you need to stimulate brain by some kind of a direct current stimulation or the magnetic stimulation, and then you record the activity of the brain and use some kind of a compression algorithms to then estimate the complexity of the response. So it turns out that the individuals that are conscious, this response to this initial stimulations through the skull, the response is more complex than in people in vegetative state. And the interesting thing that when it was tested in the patients with minimal conscience and in vegetative state, it found that in people with minimal conscience, so they served as positive controls of sort, it showed that this technique allows to predict consciousness most of the time. So they had only 2 false negatives when they thought that person is unconscious while he was conscious, in fact, out of 38 people that they tested. And another interesting thing that when they were testing people in vegetative state, so they could not have any kind of response. So these people, they cannot talk to you, they do not show anything at all. So not really know what is inside of their brain. So they found that using this technique, they could detect 9 people that were possibly conscious. So they could be locked in within their brains without any connection with the outside world, but they could be conscious inside without any ability to interact with the rest of the world. And to finish our little talk about consciousness, I have to say that there are right now 2 fundamental theories of consciousness. So while we have a working technique that can somehow estimate consciousness, people are trying to create some kind of a fundamental theory. So to understand what really defines the consciousness of a system. And these 2 theories are global workspace theory and the integrated information theory. So I will not be able to explain them to you, but now you have a reference, please. you can have a look it up after the lecture. But the main idea here is that in case of global workspace theory, this theory is explained by the theater metaphor. So there is some kind of a common workspace that interacts with a lot of stuff that is unconscious. So, but there is some kind of a place, like a spotlight in theater, where most of the action happens. So, and this action, it is influenced by some stuff that is unconscious. So there are a lot of functions that affect this particular part of activity, but they are not in the spotlight. And so for a system to be really conscious, it has to have this type of a place where systems can interact with each other. And as for integrated information theory, is that this one is much harder to explain because it operates with some kind of a measure. It's called phi, and this measure can be calculated for different systems. And the higher this phi measure is, the more conscious the system is. So human brain is really huge, and for some other stuff, it can be calculated. Some layouts of neural networks can be calculated, and the lower it is, the less conscious the system is. So, and as for comparison, it turns out that this one on the right, I don't think there are experiments that can be devised right now to test it, really. And as for this one, I think there are some experimental setups that can be used to check whether it's true or not. All right, so let's talk a bit more about some techniques that can be used to assess the neural activity with disregarding the consciousness. So, and for this, we'll go deeper into understanding of very small details of brain functioning. And the next logical step would be to place some kind of detecting electrode under the skull. So directly on the brain. And for this, you can use the electrocorticography, where electrodes are placed directly on the brain. And it turns out that, of course, skull no longer interferes and you have an improved signal recording. But the downside is that you have to physically implant it within the brain. So it's not exactly user-friendly. And it is justified in some cases when you have some kind of a neural pathology and you introduce this type of electrode to counteract epilepsy, for instance. But maybe you would not be able to do this type of intervention for regular normal people because it is highly invasive, as you can see on this picture. But the upside from this is that in turn you get a very detailed information from a specific part of the brain where you install this kind of electrode. And it turns out that this type of recording allows you to get very sophisticated stuff directly controlled by the brain. So if you place this array of electrodes on the motor cortex, you can get a prosthetic hand. So with the application of some kind of machine learning algorithms to learn what brain exactly wants. So you need to decode it first, but then this signal can be used to control the prosthetic arm. And well, maybe it is important for people that were amputated. So for them, it's a very natural, would be a very natural way to control their prosthetic hand. To go even further, even deeper in the spatial resolution, there is a technique that is called the local field potential. And in this case, the array of electrodes is even smaller. Once again, it is embedded in the brain, but this technique boasts a very high spatial resolution. So this very small array array of electrodes, smaller than a coin, is inserted directly in the brain. And well, this way you get an even more specific recording from neurons. You can go even deeper and start recording activity of a single cell. And for this, there are a variety of methods. One of them is to use a patch clamp to attach pipette directly to cell surface to probe its electrical activity. And there are some different setups for this patch clamp, and you can even record activity of a single ion channel, for instance. But you can also record it for a whole cell. Of course, this type of manipulation is highly invasive, and maybe it's not very practical to record only one cell at a time. But in some cases, you would want to know this kind of information. And also the deepest possible level when you can record neural activity is to insert an electrode directly inside the cell. So in this case, you get a very precise information as to how the cell functions. So let's go to another part of what we want to know about the brain, and this part is about the connectivity. So we know that neural system is characterized by conductance. So in some cases, we would want to know what connections are between what connections exist between different parts of the brain. And the reasoning for this might be that in some cases, at least, the structure of the system defines its functionality. So if we look at the wing, you can probably somehow think that it is used for flight. And if you look for a leg, you can detect that it is used for walking. So in case of brain, if you are looking at a specific pattern of connections, you can maybe analyze the function of the system. And of course, people are employing different techniques to study connectivity. Well, first of all, in different setups, fMRI can be used for finding connectivity, and in these cases, not exactly fMRI, and a different kind of magnetoresonant imaging can be used for finding connections, and it uses a different feature of the neural system, is that water diffusion inside the neural system is disrupted by myelin sheath that are developing the neural tract. So by finding this type of barriers, you can then visualize connectivity on a great scale. And also you can use different ions that are only transported along axons, for instance, of neurons. And in this case, once again, you can see where these axons lie. So if you add manganese ions that get into the cell and are transported via the axonal transport, you can highlight some kind of anatomical features, and you can trace neurons. And of course, you can also try to somehow find connectivity by implying that regions that function together have to be connected with each other. So if you find some correlations between activity of different brain regions, you can deduct from this information the connectivity between these regions of the brain. So maybe a very simple thing to think, but it is also used quite often. But as for very high resolution techniques, we can also discuss the finding the connectivity with the revolution of a single cell. And for this, you can use techniques that are called neural tracing, and it can be both retrograde from synapse to body and anterograde from body to synapse. So in some cases, in very specific cases, you would want to know where cell projects, so what does it innervate. And it turns out that nature has tools for precisely this type of problem, because there are some viruses like rabies virus that are neurotropic, and they can travel along synapses. So rabies virus can be used for neural tracing because of the way it functions during the pathogenesis of rabies. So when a dog that has rabies bites you, first of all, this virus travels from muscle to the nerve that innervates this muscle in this synapse, neuromuscular junction, and then it travels along the neural pathway all the way up to the brain. So this virus can somehow jump along these neurons that are connected to each other, these synapses. And you can utilize this feature to trace the projections of neurons. And yeah, so there are some very specific histological features of this rabies viruses, some bodies that are used to replicate and to transcribe rabies-specific molecules. So, and the way it works is that you need to add to the rabies virus, to use it for neural tracing, you need to add a payload that can be detected by you. So it might be some kind of fluorescent marker, but also you can utilize whatever payload you would want. And the way you introduce it into the rabies virus is that you need to substitute one of the proteins of the virus itself with this useful payload. And it also serves a double purpose, is that this virus, without this protein, without the surface protein, can no longer propagate. So if you inject it, this type of virus, it can only multiply within one cell, and you'll not be able to travel along the neural system, and it can be used as this monosynaptic tracer. So the way it works is that, first of all, you will deliver G protein, the envelope protein, and the rabies virus genome into the cell. their virions are assembled that contain normal surface proteins, so they can travel along synapses to other cells. But the genome that we introduced into the cell simultaneously is not encoding this same protein, G protein. And when virions are assembled, they can only travel to, so these virions are normally coated and they can infect neighboring cells, but when they infect these cells, the genome was faulty and they can no longer produce normal virions. So cells that are lying further beyond, they cannot be infected. So you only see highlighted the first order connections of the initially infected cell. So in this case, you can recreate the neural networks in a very small scale, starting from one cell, for instance. But this technique is not exactly high throughput, because you need to do these injections with the singular cells, and so for large scale reconstruction of connectomes, it's not exactly practical. So the way you can do this, the way you can reconstruct connectome in a more large scale approach, is that you can utilize the reconstruction of connectome from thin tissue slices. So you can just take a brain, if it's a very small brain, like a mouse brain, or maybe some invertebrate brain, and then you slice it like salami in very small pieces. You scan all of them with a scanning electron microscopy, and then you align all of these small photographs into a 3D structure. And you can also do this in an automated way. So there are algorithms right now that can reconstruct connectivity from stacks of photographs, and you can get ideally the reconstructed structure of neurons inside the some kind of a neural system. And so yeah, that's how this process goes. You need to align all these photographs, and in the end, hopefully you see all of the axons and dendrites from neurons, and you can reconstruct some kind of box also within the neural systems. And this type of work has been done for some organisms. So for some organisms, it is easier to do because, for instance, in Cineraptis elegans, a small worm, it only has 302 neurons. So it is fairly simple to find all of them inside this very small body. So, and this type of connectome for worm was in fact created in 80s. But a very recent work has done the same task, but they also, so there are two different sexes of this worm, hermaphrodite and male worms. So in this particular paper, authors recreated connectomes for both of these sexes, and they also analyzed differences between the connectivity in these worms, and they tried to find features that are maybe specific for males and hermaphrodites. And also they, of course, did all the corrections, because the previous work was very dated and maybe they were missing some kind of connection. So, and well, it turned out that worm connectome is already quite complex. So despite the fact that there are only 300 cells, there are quite a lot of connections, and for multitude of functions, like in this case, here's the network of all of the neurons that are implicated in muscular movement. And it turns out that out of 82 types of interneurons, 60 types of interneurons are implicated in this innervation of muscles. So this network is a mess of sorts. So it is hard to analyze. And authors also say that if you miss even a small part of these connections, you may not be able to analyze the behavior of the system as a whole. And also they found some kind of specificities between different types of worms. So it turned out that despite a very small size of this neural system, there are some very specific there is some very specific behavior between types of worms, and they also correspond to different types of connectivities. So there is something different between these worms. Well, worm connectome may be not so exciting for us because well, the neural system is not like ours. We have slightly more neurons. So of course we are maybe more interested in connectomes of creatures that are more resemblant to us. So, and of course people are also trying to do this. So in this case, very recently it was reported that people created the full connectome of a hemibrain of a fruit fly. So this neural system is slightly more complex. They have 25,000 neurons with an estimated 20 million connections between them. And for this large-scale work that was done in 12 years, Google even created a special algorithm for interpreting this connectomics data. So they created a special type of neural net that can color all of these parts of cells to then recreate the whole, the connectome within the Drosophila brain. And yeah, so right now, it's not exactly clear what to use this information for, because a lot of information is lacking right now. So we don't know the full connectome of human, for instance. So the applications of this type of information are not clear, but it was also so with the genome sequencing when it only started. So people didn't really know how do we map genome, how do we use this type of information. So maybe it's a different type of question. So maybe when we have the technology to reconstruct connectome very fast and very reliably, we can also find the application for this type of information. And of course, for humans, the species we mostly like and we are most interested in, there is also a specific project, so a human connectome project that aims to reconstruct the whole connectome of the human brain. Of course, it is very hard. So right now, we are not even close to completing this type of project. In parallel, there are some projects that aim to reconstruct connectomes of singing birds, of small parts of the mouth brain, but the implications for this, if you get this type of information, maybe it will be useful for connectomes of during your lifetime, of difference of connectomes in diseased in normal states, and so on and so forth. So we are talking about the analytic part of the neural technology. So now let's talk a bit about how do you control the neural activity. So how do you apply the engineering to this? How do you somehow change states of neural systems? So, and first of all, some techniques like recording of electrical activity from electrodes, it gives a very natural way to control the activity of these neurons, because when you're recording with an electrode in this very invasive manner, you can also provide some kind of you can also excite the cell, for instance, and you can activate select neural populations with these electrodes. And in this way, you can manipulate the activity. But there are some other techniques. Yeah, like you can use, the electrodes, first of all, they have the applications already, because they can be used for deep brain stimulations for some diseases like Parkinson's disease, and it's even approved right now as the normal therapy for Parkinson's disease. But there are some other ways you can affect the functioning of neural systems, and one of these it utilizes genetic modification, and it couples them with the control of functioning of cells with light signals. And the functionality here is triggered by the fact that photoreceptor interacts with the photon of a specific wavelength. So by doing this, by projecting light to the brain of the mouse, you can control its behavior possibly. So if you understand what you're activating. And the way it works is that, well, to excite the neural cell, you need to activate some ion channels on its surface. And so for this to work, you need to find some light activated ion channels. And it turns out that there are actually ion channels that can be activated by light and can be used for this. And well, first of all, of course, we know that there are some proteins that are activated by light even within our organism. And these proteins are taking part in our vision. And some of these types, they are present in vertebrates. And so they are expressed in photoreceptors. And with their help, we perceive light. But they are not exactly useful for these particular applications. And it turned out that there are proteins that exactly answer to our call. They provide the solution for this problem. So there are proteins that are ion channels, and they are responsive to light, and they are called channel rhodopsins, and they are light-sensitive cation channels. And when they were originally found in algae, and when you activate them with light, they depolarize the membrane. So this way you can excite the neuron. So here's the structure of this channel rhodopsins. And it turns out that channel rhodopsins, the initial ones they found, are permeable for different cations, and they are not permeable for anions that can provide a very specific activity when you activate them. So you depolarize the membrane. And also they are coupled with the rectifying potassium channels. In initial studies of these channel rhodopsins, they were characterized by expression in embryos, and it turned out that you see exactly the activity that you would expect from certain limited genomes. So when you activate them with light, it leads to action potential. So you expect it from the depolarization. And if you increase the light pulse duration frequency, you see some sub-threshold activations. And they're also expressed in a very expressed in an expected way. And it turned out that they were enough to control some things that were to affect things that were controlled by the neural system. So the light induction of this initial channel rhodopsin, it caused electrical activity in the motor neurons that control the action of limbs. Of course, right now, a lot of time has passed from the initial discovery of channel rhodopsins, and now we have some alternatives as to light-activated channels. So right now, there is a multitude of proteins that can be utilized in optogenetics, and there are some other principles that underpin this type of function. For instance, there are calcium ion channels that are activated by light, but they function with another type of light-sensitive protein that came from plants, from cry protein. And also they have also the ability to activate some parts of the brain, and if you introduce them to very specific neurons, you can use it to reinforce contextual memories, as in this study. So yeah, it can also be used to provide a very specific change in neural activity. And yeah, like I mentioned, right now there are a lot of types of channels for very different applications in optogenetics. So you can use them for inhibition, for excitation. You have proteins with different kinetics, so you can use some of them for very fast activations, some of them for very slow and gradual activations, and yeah, they are characterized by very different times of response to the initial light activation. Yeah, and very recently, one of the creator of regional optogenetics technique, Karl Desserot, he published another interesting paper about that addresses a very prominent problem in the field of optogenetics, is the fact that normally to activate cells within the brain with the optogenetics, you need to introduce some kind of a optic fiber to conduct the light from the outside to the neuron populations. So it may not be exactly useful in most of the cases. So you would want some kind of a non-invasive procedure for optogenetics to be truly useful medical conditions, for instance. So, and it turns out that people found recently a version of channel rhodopsin, it's called CHR9, that is very sensitive and it allows transcranial stimulations of deep brain areas. So you can just make skull thinner in some place, and you can project light through the skull with a fiber. And turns out that if you introduce this channel rhodopsin in a very deep brain area, it will still be activated, despite the fact that there are a lot of there is a thick layer of tissue here, and well, it will all reflect and disperse light. So despite the fact that all of this tissue is present here, it will still be activated. Of course, optogenetics, in the definition that I mentioned several slides previously, it is not restricted to ion channels. So other proteins can also be light sensitive, and it is utilized sometimes for other applications. Like, for instance, there are some setups that allow to activate protein interactions when you irradiate your tissues with the ultraviolet light. And it functions because of the protein domain from the protein UVR8 from plants, once again. And it can be used also to regulate transcriptional activity. And also right now there are ways to couple light activations with genome editing. So you can have light controlled genetic modifications in genome, and to not leave it up to the case to have a really controlled interaction with the brain, with the genome. It doesn't matter what types of cell here. So yeah, there are some ways you can affect the neural system, and one of the applications of this type of the interaction with the neuron system is the neural interfaces. And the way it can be used is to, the most natural application would be to provide the biofeedback, is to control some normally involuntary functions. And it is immediately useful for treatment of headaches, for pain management, but it can also be used to provide the muscle control, the control of mindfulness, provide the breathing control. So neural interfaces right from the start have a lot of applications for well-being of individuals. And it turns out that neural interfaces are already in use right now. So it's not something really fantastic, not something from sci-fi literature, because we already have cochlear implants, and they are a very specific type of neural interfaces. So the way it works is that, well, if people are not able to hear naturally, this cochlear implant, it receives the signal to the microphone, and it transmitted to the auditory nerve. And here you can see the QR code on this slide. You can look it up later, but it's a YouTube recording of how the sounds are to people with this cochlear implants. So they are slightly disrupted, but it is possible to understand at least what you're hearing at any particular moment. And there is an interesting feature about this cochlear implant is that is the way the sound is encoded in the ear, because in this case it is quite simple to understand what do you need to do for this implant. They encode different frequencies of sound. So when you receive them through the microphone, you understand where exactly you need to excite neurons. So it is a fairly well understood part of the human brain. So in this case, it is fairly simple to decode, to encode and to decode signal for this neural interface. So as for other senses, right now it's not so bright, but there are some promises in well, as in case of vision, there are techniques that measure electrical responses from photoreceptors, from bipolar and ganglion cells within the retina. And so right now they're mostly used as a diagnostic tool, but potentially it might be interesting to also use this type of interface to restore vision in some individuals. So tissue engineering is not the only way you can do this. Yeah, and as for more complicated neural interfaces, right now there are a lot of studies where you can utilize neural interface for different applications. Like this one, when neural interface was used to control an animal, when the signal from the person, the operator with the electroencephalograph was transmitted to a trained rat that knew how to respond to different signals sent from this operator. So this rat can turn left or right in the cases. So the operator knows the layout of the labyrinth, and it can transmit the instructions as where to this rat should go. And also in this paper there is a very funny video when you can when the person controls this rat traversing the maze, the 3D maze. And of course, when talking about neural interfaces, I have to address the elephant in the room, and I have to talk about the Neuralink, one of the new companies that aim at creating commercial neural interfaces and very large-scale neural interfaces. And the most interesting feature here about the Neuralink is that it employs many electrodes at the same time, and it also involves the robotic surgery for the introduction of these arrays of electrodes. So this surgery platform here is on the picture from the original paper by Neuralink. And the advantage of this type of robotic platform is that it can introduce very small electrodes with a very high precision inside the. So in this case, it's a replica of the brain, but it can introduce the small electrodes into the brain with a very high precision. And it is really important for the introduction of these types of electrodes, because the less damage you cause to the neural tissues, the more efficient is the function of this electrode. So if you damage a lot of tissue, the tissue will answer by degrading the electrodes a lot of times. So the chronically implanted electrodes, they have a lot of problems with this type of stuff. And if you are not very cautious when introducing them, you may have non-functional electrodes in a very short time. So yeah, that's one advantage. And another advantage that is this device is introduced inside the brain. It is really small. So it consists of the link, a device that is in fact introduced under the skull. So on the surface of the brain. It has multiple neural threads that are introduced within neurons, and they are introduced to the robotic surgery. And it has a charger that can be used to, it connects to the implant wirelessly and can be used to charge the battery from the outside. So here's what the connection of electrodes to the rat brain looked like a year ago. And they utilized this USB-C connection, and they had more than 3,000 of channels. But yeah, and every channel can also be used for stimulation. So you're not really restricted to recording. All of this can be used to alter the neural activity at the same time. And as for this year, in this August, we had another presentation. And this year, the Neuralink chip was introduced into a pig named Gertrude, and this time they had more than 1,000 channels with the real-time recording, and also, well, they recorded a lot of stuff, how this pig walked on a treadmill, and they reconstructed the position of joints in the real time from neural signals, and it turned out, yeah, you can see there are some small details here, but the predicted position of joints was very similar to the real position of joints when the pig was walking on the treadmill. So you can get a lot from this reading of multiple electrodes. And here's the QR code of when this pig was, well, there was a handler in the cage with this pig, and when it bumped its snout, you could see the, well, some neural signal from this Neuralink chip. And right now we know that Neuralink applied for human trials, so possibly in some near future we might see something like this when we have a Neuralink app that allows you to remotely control your iPhone to use it for, I don't know, for typing of characters on the keyboard for different types of games, and if it gets hacked, you may get something like this. But yeah, possibly you may see something like this in the near future. So, and of course, well, aside from this very specific example of neural interfaces, right now there are some neural interfaces that are already in use. So they are already approved for human usage. And here you can see the very specific neural interface for tetraplegic patients that can restore them some kind of a functionality. And the very strange example, and the last for this lecture, is the strange setup when you can connect brain to brain with the usage of a computer as an intermediate. And it was done in this particular paper where people used electroencephalography and transcranial magnetic stimulations to connect 3 people. And the way it worked is that these 3 people played a game, and they had 2 senders. They had the full information, and they had a receiver that had to, well, perform an action. So it was a Tetris-like game. At senders, they saw what they had to do, either to turn piece or not, and the receiver had to decide, does it, well, from the information that it got from the stimulation, does it decide to rotate the figure or not? So, and they saw that in some cases, even with the disruption of the signal, you were able to play the game better than without any additional information. So it turned out, so in some cases, especially in good senders, the game was, well, much easier for the receiver. So thank you, everyone. If you have any questions, you feel free to ask them right now. All right. Thank you once again, everyone, for your attendance. Yeah. Goodbye.